Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today we're talking about topic 3.1, enzymes. So what will we learn today? For today's lesson, we're going to look at what is the role of enzymes, how is enzyme structure related to enzyme function, what determines whether a substrate can interact with an enzyme, and finally, what happens to an enzyme after the completion of a reaction. So let's dive in. Enzymes are biological catalysts that speed up biochemical reactions. Most enzymes are proteins, so they have a tertiary shape that must be maintained for functionality. They have a region called an active site. So if you take a look at the picture, you'll notice two different enzymes, two different active sites. A molecule that can interact with an enzyme is called a substrate. Enzymes have an active site, specifically that interacts with substrates. That active site has a unique shape and size. It can have a chemical charge or charges or not. The physical and chemical properties of the substrate must be compatible to the active site. And then finally, slight changes can occur in the active site to align with the substrate. Enzyme names often indicate the substrate or chemical reaction involved. These names often end in ASE. For an example, sucrase. Sucrase is an enzyme that digests sucrose. Enzymes are reusable. They are not chemically changed by the reaction. Cells typically maintain a specific enzyme concentration. And finally, enzymes can facilitate synthesis or digestion reactions. So that is shown in the picture on your screen. So let's practice. For today's practice, the skill focus is concept explanation. So you're going to explain biological concepts and/or processes. So let's take a look at the question. What I would like you to do for your question is I would like you to pause your video. First, read the prompt. Look for the bold-faced word, that is the task word, and then ask yourself, what are you being asked to do? Then reread the prompt and formulate your answer. Once you have your answer, push play again. Welcome back. The question asked you to look at the study of the rate at which salivary amylase hydrolyzes amylose into glucose and best explain the chemical interaction between amylose and salivary amylase. Your answer should have been D. Amylase active sites have similar chemical properties to amylose, allowing glucose products to form. It best explains the chemical interaction between amylose and salivary amylase. Amylase has a polar active site, and amylose is a polar carbohydrate polymer, so they're compatible. So what should you take away from this lesson? First, enzymes speed up chemical reactions by lowering activation energy requirements. The structural characteristics of an enzyme makes the enzyme very reaction specific. The shape and charge of the substrate must be compatible with the active site of an enzyme for a reaction to occur. And finally, enzymes are not consumed by the reaction. Enzymes are reused. Well, I hope today's video was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is enzymes. So what will we learn today? For today's lesson, we're going to look at two things. How do enzymes facilitate chemical reactions in cells? And what is activation energy in a chemical reaction? So let's dive in. First, enzymes are biological catalysts, typically proteins that speed up biochemical reactions. Enzyme structure is very specific, resulting in each enzyme only facilitating one type of reaction. Enzymes can facilitate synthesis or digestion reactions. The example on the screen is an example of an enzyme-mediated digestion reaction. All biochemical reactions require an initial starting energy called activation energy, as shown on the chart. Some reactions result in a net release of energy, and some reactions result in a net absorption of energy. Typically, reactions resulting in a net release of energy require less activation energy compared to reactions resulting in net absorption of energy. So what do enzymes do? Enzymes lower the activation energy requirement of all enzyme-mediated reactions, thus accelerating the rate of reactions. So if you look at the chart, you can compare the amount of activation energy with an enzyme and without an enzyme. Let's take a look at an example. For this example, this is a stop motion video using pool noodles to model an enzyme substrate interaction. The first video is of a synthesis reaction where the enzyme lowers an activation energy, and the second one is of a digestion reaction. All right, so now let's practice. For today's practice, we're looking at the skill focus of questions and methods. You're going to identify experimental procedures that are aligned to the question, including identifying appropriate controls. So let's take a look at the question. The first thing I would like you to do is pause your video. Then you're going to examine the data table and ask yourself what you see and what it means. Then read the prompt and look for the bold-faced word, that's the task verb, and ask yourself, what are you being asked to do? Finally, reread the prompt and formulate your answer. Once you have your answer formulated, push play. Welcome back. For this question, you were looking at an experiment that involved studying the actions of the enzyme catalase on hydrogen peroxide. Your answer should have been A, which was placing a catalase soaked disc in a beaker of water. The other three choices introduced a new variable to the experiment and are not controls. So what should you take away from today's lesson? First, enzymes are biological catalysts that facilitate chemical reactions in cells by lowering activation energy requirements. Second, activation energy is the initial energy required for a reaction to occur. Well, I hope today's video was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is enzymes. So what will we learn today? In today's lesson, we're going to go over what are the different types of control groups, what is the use of each type of control group, and then how are control groups different from control variables? So let's dive in. A controlled experiment is a scientific investigation. There are two types of tests set up in a controlled experiment. First, you have the control test or control group. It generates data under conditions with no treatment or no manipulation. It also generates data under normal or unchanged conditions, and it is considered the baseline data. The other group is the experimental test or group. It generates data under abnormal or unknown conditions. It also generates data under treated or manipulated conditions. Test results often compared with control test results to help determine possible impacts of a treatment or manipulation. A control group is used as a standard for comparison. One type of control group is a negative control. The negative control is not exposed to the experimental treatment or any treatment known to have an effect. A positive control group is exposed to a treatment that has a known effect. It is not exposed to the experimental treatment. Both types of controls can be used to validate experimental procedures. Controlled variables are aspects of an experiment that could be changed but are intentionally not changed. They are important to help isolate and identify the impact of intentional change or treatment. Only variables known to have an impact should be considered as possible controlled variables. Control variables are also known as constants. So let's look at an example. For this example, I would like you to pause your video and then first examine the data table and ask yourself what you see and what it means. Then read the prompt and look for the bold-faced word, that's the task verb, and then ask yourself, what are you being asked to do? Then reread the prompt and formulate your answer. Once you have your answer formulated, push play. Welcome back. For this question, you were asked to identify the control group of the investigation that involved looking at the effect of various temperatures on the enzyme activity of peroxidase. Your answer should have been the control group is equal to 37 degrees Celsius. This test represents the normal temperatures for human cells. This would be an example of a positive control. It's exposed to a treatment that has a known effect. Now let's practice. For this practice, the skill focus is going to be questions and methods. You're going to look at identifying experimental procedures that are aligned to the question, including justifying appropriate controls. So let's take a look at the practice. So again, I want you to pause your video, and then I want you to examine the data table and ask yourself what you see and what it means. Then read the prompt and look for the bold-faced words that are the task verbs. And then ask yourself, what are you being asked to do? Then reread the prompt and formulate your answer. Once you have your answer formulated, push play again. Welcome back. For this question, you were asked to propose a negative control for the experiment that looked at the effect of pH on the enzyme polyphenol oxidase and justified the purpose of the control. Your answer should be one of three different negative controls. One, reaction without an enzyme, which confirms the enzyme is not necessary. Another choice would be reaction with denatured enzyme, which confirms that active enzymes are necessary. And then finally, reaction under anaerobic conditions, meaning no oxygen, and it confirms oxygen is necessary for the reaction. So what should you take away from this lesson? First, the two types of control groups are negative and positive control groups. Positive control groups are mainly used to confirm a known effect. Negative control groups are mainly used to confirm results in the absence of any kind of treatment. Results from control tests are used as baseline data. And finally, control variables are aspects of an experiment left unchanged across all tests to avoid additional impacts on results. Well, I hope today's video was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 3.2, environmental impacts on enzyme function. So what will we learn? In today's lesson, we're going to look at what does it mean for an enzyme to be denatured? How do changes in pH or temperature affect enzyme activity? How do changes in enzyme concentration affect the rate of reactions? How do changes in substrate and product concentrations affect the rate of reactions? And finally, how do competitive and non-competitive inhibitors affect enzyme activity? So let's dive in. Enzymes have a unique functional 3D shape known as the conformational shape or tertiary structure. Changes in the conformational shape of the enzyme equals denaturation. Changes in the environmental temperature can lead to denaturation. Changes in environmental pH can lead to denaturation as well. Enzyme denaturation is typically irreversible, and the catalytic ability of the enzyme is lost or severely decreased. However, in some cases, enzyme denaturation is reversible, allowing the enzyme to regain catalytic activity. So what does it mean by optimum temperatures? Well, optimum temperature is a range in which enzyme-mediated reactions occur the fastest. Reaction rates change when optimum temperatures aren't maintained. When there's an environmental increase in temperature, there's an initial increase in reaction rate. This causes an increased speed of molecular movement and an increased frequency of enzyme substrate collisions. Temperatures increases outside of the optimum range result in enzyme denaturation. When the environmental temperature decreases, it generally slows down the reaction rate, as shown on the chart. It leads to decreased frequency of enzyme substrate collisions. However, it does not disrupt enzyme structure, so no denaturation occurs as the temperature decreases. So what about pH? pH measures the concentration of hydrogen ions in a solution. It is measured on a logarithmic scale. A logarithmic scale, or log scale, is a way of displaying numerical data over a very wide range of values in a compact way. So, small changes in pH values equate to large shifts in hydrogen ion concentrations. So, for example, a pH of 6 has 10 times more hydrogen ions in solution compared to pH 7. So what about optimum pH? Well, it is a range in which enzyme-mediated reactions occur the fastest. Changing the pH outside of this range will either slow or stop enzyme activity. Enzyme denaturation can occur as a result of increases and decreases outside of the optimum range. Changes in hydrogen ion concentrations can disrupt hydrogen bond interactions that help maintain enzyme structure. So how do the concentrations of substrate and products affect reaction rate? Well, an initial increase in substrate concentration increased reaction rate. More substrates mean more opportunities to collide with enzymes. Substrate saturation will eventually occur, as you can see on the chart. This results in no further increase in rate. The reaction rate will remain constant if saturation levels are maintained. However, an increased concentration of products decrease the opportunity for addition of substrate. Matter takes up space, so the more product in the area means lower chance of an enzyme substrate collision, and that will slow the reaction rate as shown on the chart. So how does enzyme concentration impact reaction rate? Well, first, changes in enzyme concentration can also impact reaction rate. Less enzyme equals slower reaction rate. There is a less opportunity for substrates to collide with active sites. However, more enzyme equals faster reaction rate. There is more opportunities for substrates to collide with active sites. So, competitive inhibitors. They are molecules that can bind reversibly or irreversibly to the active site of the enzyme. They compete with the normal substrate for the enzyme's active site. If an inhibitor concentrations exceed the substrate concentration, the reactions will slow down. If inhibitor concentrations are considerably lower than the substrate concentration, reactions can proceed normally. If an inhibitor binding is irreversible, the enzyme function will be preventable. However, if an inhibitor binds reversibly, the enzyme can regain function once the inhibitor detaches from the active site. Enzymes can have regions other than the active site to which molecules can bind. These are called allosteric sites. Non-competitive inhibitors do not bind to the active site. They bind to the allosteric site. Binding causes conformational shape change to the active site, and the binding prevents the enzyme function because the active site is no longer available. The reaction rate decreases, and increasing the substrate cannot prevent effects of non-competitive inhibiting binding. So now it's time to practice. The skill focus today is argumentation. We're going to predict the cause or effect of a change in or disruption to one or more components in a biological system based on data. So let's take a look at the question. So for this question, I would like you to push pause and then first look at the data table and ask yourself, what is it telling you? Next, read the prompt and look for the bold-faced words, which are your task words. And then ask yourself, what is the question asking you to do? Finally, reread the prompt and then answer the question. When you have your question answered, push play again. Welcome back. For today's question, you were looking at making a prediction of the average time it would take for catalase to make a disc placed in 1% hydrogen peroxide solution float if the catalase was boiled for 10 minutes before adding to the solution. Your answer should involve that the disc would never float, or it would take more than 30 seconds. So what should you take away from today's lesson? Well, first, denaturation of an enzyme occurs when the conformation protein structure is disruptive, eliminating the ability to catalyze reactions. Changes in pH outside of that optimum pH range in either direction will result in decreased enzyme activity and eventually lead to denaturation of the enzyme. Increasing temperatures outside of the optimum range will initially cause an increase in reaction rates, with continued increasing resulting in denaturing of the enzyme. Decreased temperatures outside of the optimum range, however, will result in a slowed reaction rate, but not denaturing of the enzyme. Increased enzyme concentration, increased reaction rate. Decreased enzyme concentration leads to decreased reaction rates. Increasing the substrate concentration will initially increase the reaction rate, but substrate saturation will not result in continued increase in reaction rate. And then the reaction rate will remain constant if the substrate saturated levels are maintained. For competitive inhibitors, they can bind reversibly and irreversibly to active sites, potentially altering reaction rates. Finally, non-competitive inhibitors can bind to allosteric sites, decreasing the catalytic capability of enzymes. Well, I hope this lesson was helpful. Until next time, thank you.
Good morning, good afternoon, good evening, AP Biology. My name is Chris Mansour, and I'm an AP Biology teacher from Tiffin, Ohio, and I teach high school biology at Tiffin Columbian High School. And I'd like to welcome you to topic 3.3, cellular energy. This is daily video 1, and this lesson will describe the role of energy in living organisms like yourself. And we're going to focus on a skill today, and that skill will be on how to provide reasoning to justify a claim. Justifying a claim is a very important skill you need to have to be successful in the AP Bio exam in May. And so what will we learn? Always a good question. We need to have some kind of objective. So what will we learn? What do living things use chemical energy for? So maybe pause the video here and jot down some ideas, but think about as you're sitting here, what are you using chemical energy for? Is it to try to pay attention? Is it to write down some notes, right? And how is energy efficiently transferred in a biological pathway? Efficiently is the key. We want to be able to conserve, and we want to be able to be efficient as possible, because as you're going to find out, we do waste a lot of energy. So all living systems require a constant input of energy, and sunlight is that main energy input for a living system. As you can see from that diagram, that light energy from the sun is going to be hitting that chloroplast of that autotroph. And as we hopefully know, autotrophs capture energy from physical sources like sunlight or chemical sources and transform that energy into an energy source usable by all cells. It would be nice if we could just go out in the sun and photosynthesize. It doesn't work that way. And so we do need sunlight for autotrophs. But during every energy transformation process, some energy is unusable and often lost as heat. And so as I said, as we're sitting here now, we are giving off unusable energy in the form of heat. So when we're thinking of energy, we have to understand that life requires a highly ordered system. And highly ordered is important, and it does not violate the second law of thermodynamics. There are two of them, but we're only going to focus on the second law of thermodynamics. Every energy transfer increases the disorder of the universe. I like to think of this as some Dixie cups stacked up. I knock them over with my hand, and I've just created disorder. Living cells are not at equilibrium. If you think of equilibrium, you're thinking of like biological balance. There's a constant flow of materials in and out of the cell. Think of it like when you eat. You're taking food in, waste is coming out, but there has to be a constant flow of materials for you to survive. Same idea, living cells are not at equilibrium. They're taking in materials in, and they're releasing materials. And cells manage energy resource by energy coupling. So energy coupling, that's where an energy releasing process drives an energy storing process. So energy is released and that drives an energy storing process. And also when we talk about energy, we need to talk about pathways because pathways is how energy is transferred. So pathways in biological systems are sequential. Within a chemical pathway, like I have here in blue, the product of one reaction can serve as a reactant in a subsequent reaction. The sequential reactions allow for more controlled and efficient transfer of energy. So we've got a starting molecule A, those are your reactants. We have linking reaction B to products B. So there's one step in that biological pathway. The products of B will eventually become the reactants for linking reaction 2 to make product C. Then product C will become the reactants for reaction 3 to make product D. The big idea here is that what you start out with A, those reactants are going to be much different than those products at D. But by doing this in a series of small steps, it allows for more controlled and efficient transfer of energy. So let's do some practice. Practice makes perfect. You guys are getting ready for that exam in May. I know it's September here right now. Your teachers are working hard, but we're going to focus on argumentation. Provide reasoning to justify a claim by connecting evidence to biological theories. That's important, is this reasoning part. So skill, practice, argumentation. Again, a researcher claims that the incorporation of carbon dioxide into organic molecules during photosynthesis does not violate the second law of thermodynamics. Which of the following statements best, key point, best helps justify the researcher's claim? I'm not going to read all those options to you. I'm going to pause the video so that you can maybe talk amongst your classmates, maybe in a Zoom chat, whoever is with you at home. So go ahead, pause the video, and I'll be back here in a second to get your answers. Okay, and we're back. Okay, Sue, who has letter A? Any A's? I'm in here by myself, my classroom. B, okay. C, couple of you. D, the D's have it, so good job. Give yourself a little pat on the back or whatnot. The total system that includes photosynthetic organisms and the sun becomes less ordered over time. The key is less ordered, tying that back in to the fact that the researcher was looking for something that does not violate the second law of thermodynamics. So what should we take away today? Key takeaways: 1, living things like yourself use chemical energy stored in molecular bonds of macromolecules like carbohydrates and ATP to perform necessary life functions. 2, pathways in biological systems are sequential. That's the key, going back a couple slides to that pathway, to allow for a more controlled and efficient transfer of energy. So again, I want to thank you. I know you're working hard. I know your AP teachers are working hard, and I appreciate the time you spent with me. So again, my name is Chris Mansour. I'm signing off from room 102 at Tiffin Columbian High School, and have a great day, and I'll see you in a future video. Good morning, good afternoon, good evening, AP Bio. I'm back for another video. This is topic 3.4, photosynthesis. My name is Chris Mansour, and I'm coming to you from Tiffin Columbian High School, room 102. In daily video 1, this lesson will explain how the process of photosynthesis allows organisms to capture and store energy. The skill that we'll focus on today is on how to support a scientific claim. And again, the scientific claim and these skills are very important for you to do well on the AP exam in May. So what will we learn? What will we learn? How do organisms capture and store energy for use in biological processes? How are the light-dependent reactions involved in the capturing and transforming of energy? What are the products of the light-dependent reactions? And how are the light-dependent reactions and the Calvin cycle reactions related? We got a lot going on, but my plan is to break it down into small steps that you'll be able to kind of understand and also come up with good questions for your teachers. So the first thing, organisms capture and store energy for use in biological processes. Photosynthesis is that biological process. That is the process that captures energy from the sun and produces sugars. And evidence supports the claim that prokaryotic photosynthesis by organisms such as cyanobacteria was responsible for the production of oxygen in the atmosphere. So these early prokaryotes were responsible for the production of oxygen in the atmosphere, which is important. And finally, photosynthetic pathways are the foundation for eukaryotic photosynthesis. And so here on this picture on the right, we have a process of photosynthesis, just kind of giving you an overview. So the light-dependent reactions, requiring light, of photosynthesis in eukaryotes involve a series of pathways. So these pathways are these small sequential steps. So light-dependent, again, dependent reactions capture light energy by using light-absorbing molecules called pigments. Think of pigments as colors, right? And so if you see my blue arrow or the arrow going in, you can see where we're pointing to where that pigment source would be. Pigments help transform light energy into chemical energy, and that chemical energy is going to be temporarily stored. So that light energy is going to get converted into chemical or transformed into chemical energy, and that chemical energy is then going to be temporarily stored in a chemical bond of a carrier molecule called NADPH. So what happens is then those light-dependent reactions, not only do they help store that energy in NADPH, but they also help facilitate ATP synthesis. And so in that blue box there, if you look, you can see where I've isolated, you can see the ATP being produced, and you can see some NADPH being produced. Just follow the arrows. And ATP and NADPH transfer stored chemical energy to power the production of organic molecules in another pathway called the Calvin cycle. And so you can kind of see a sequence: sunlight hitting the pigments, you have your ATP, your NADPH being produced, going into the Calvin cycle. So think of this as a small series of pathways. And another important part of this pathway is that oxygen is produced as a result of water hydrolysis. And so that's where the oxygen is being produced, is when that water is broken up. So let's do some practice. Lesson skill argumentation. We're going to support a claim with evidence, again, evidence from biological principles, concepts, processes, and or data. And again, these skills are important for you to do well on the AP exam. So here's our skill practice: argumentation. A researcher claims that the initial rise of oxygen in the Earth's early atmosphere, which occurred approximately 2.3 billion years ago, resulted from the metabolic activity of prokaryotic organisms. The claim is based on an interpretation of geochemical and fossil evidence represented in Figure 1. I'm not going to go over the image with you, but again, which of the following types of evidence will best support the researcher's claim? Go ahead, pause the video, talk to your friends if you're in a Zoom chat, or if you're in a classroom, or again, if you're sitting there eating breakfast with your family or whoever, maybe show them this question, see what they think. So go ahead, pause the video, and I'll be back whenever you're ready for the answer. And we're back. So hopefully you had some really lively discussion. So again, which of the following types of evidence will best support the researcher's claim? A, who's got A? Again, I'm in my classroom by myself, so I can pretend my students are here. B, maybe a couple of you. C, all right, D. Best answer is D in the green box. Evidence that cyanobacteria, if you noticed, we had talked about cyanobacteria previously, produce oxygen as a byproduct of photosynthesis. So again, that is the type of evidence that best supports the researcher's claim. So what should we take away from this video? There is a lot going on here, but here are key takeaways. Again, maybe you want to pause this and let me write down these key takeaways and go back to the video. Plants and other autotrophs use pigment to trap light energy to make organic molecules. The pigment used in the light-dependent reactions help transform light energy into chemical energy. So again, a transformation process. The chemical energy is temporarily stored in the chemical bonds of that carrier molecule called NADPH. The products of the light-dependent reactions are ATP and NADPH. If you notice, we're focusing on what the products are because the products are going to be important because they're going to eventually be used as reactants. And then ATP and NADPH are products that will be used in the Calvin cycle to produce those carbohydrates. So again, I want to thank you for your time. I know this is a lot of material. I want to thank all those AP biology teachers out there working really hard to get your students ready. And again, this is my name's Chris Mansour. I'm signing off from beautiful room 102 at Tiffin Columbian High School in the heart of Tiffin, Ohio. And good luck, and I'll see you in a future video. And good morning, good afternoon, good evening, AP Biology, and this is AP Biology Daily. My name is Chris Mansour, and I'm coming to you from room 102 at Tiffin Columbian High School, and today, topic 3.4, photosynthesis, and this is Daily Video 2. This lesson will explain how the process of photosynthesis allows organisms to capture and store energy. The skill that we're going to focus on in this video is how to support a scientific claim, which is a very important skill to have when for the AP exam that's coming up in May, even though it's only September now. So what will we learn? What is chlorophyll's role in boosting electron energy levels? And that's important because by energizing those electrons is how we can transfer energy. How do photosystem 1 and 2 work together to create a proton gradient which facilitates ATP synthesis? And how are the products of the light-dependent reaction used in the Calvin cycle? There's a lot of content in this video, so you may want to kind of pause it, go through and find some vocabulary words you might not be familiar with. So let's get to our first important point. During photosynthesis, chlorophylls absorb energy from light. And so what role do chlorophylls play in this process? So maybe pause the video, jot down some ideas. Well, what they do is the chlorophylls, they capture energy from sunlight and convert it to these high energy electrons. And so what happens to these electrons, or these chlorophyll electrons, when light absorption occurs, and what's the importance of this? Electrons will be energized. The energy from the electrons will be used to establish a proton gradient and reduce NADP plus to NADPH. So the important part there is a reduction. So photosystem 1 and photosystem 2 are our next topic, and these are embedded in the internal membrane of the chloroplast. So I have a picture there for you showing you PS2 to PS1. Again, they are placed in the membrane in the order that they were discovered. So PS1 first, PS2. And so what is a photosystem? So again, maybe think about this, pause the video. But essentially what a photosystem is, it's a light capturing unit in a chloroplast thylakoid membrane. And so I've placed a blue box around those photosystems. So you see photosystem 2 to photosystem 1. And so again, that's where that sunlight is going to be captured. And why is the hydrolysis of water, which we see underneath, necessary as it relates to photosystem 2 and the light-dependent reactions? Well, the hydrogen molecules from the splitting of that water are released into the thylakoid space, and those hydrogens are going to be used to create an electrochemical proton gradient. So often electrochemical proton gradient, if you remember, creating a concentration gradient. And so as you can see where that blue arrow just came in, there's the water, hydrolysis occurs, and we now have oxygen in those H pluses. The key point there, follow those H pluses. Next, when those electrons are transferred between molecules in a reaction, they pass through the electron transport chain. And so again, I'm going to highlight the electron transport chain for you there in blue. Again, pause the video. I always like to have my students draw pictures and maybe write captions, so it might be a good time to maybe pause the video and redraw this. But how are photosystem 2 and photosystem 1 functionally related to the electron transport chain? Great question. Well, here's the answer. PS2 and PS1 pass high energy electrons to the electron transport chain. A lot of electron terms here, I know. And so that's their function, is to pass these electrons along the line. And so I've mentioned this a couple times. What actually is an electrochemical proton gradient? So it's a difference in concentration of protons, or those hydrogen ions, those H pluses, across a membrane. As you can see, the whole point is trying to create this proton gradient. And so let's talk more about this proton gradient. So the formation of this proton gradient is linked to the synthesis of ATP, which is all the way down there at the end of my image. So photosynthesis uses a form of passive transport, if you remember, hopefully passive transport, to generate ATP from ADP. We got to make some ATP, right? And so what happens is, there's my arrow, and you can see ADP, ATP. There is that proton gradient being used to produce that. And so what is ATP synthase? We know it's an enzyme because it ends in ase, right? So synthase, it's going to synthesize. So ATP synthase is an enzyme that creates ATP when those protons pass through the enzyme. So again, if you look at that picture there, those H plus are going to pass through, going to produce some ATP. And so finally, our last main idea, though, is the energy captured in the light powers the production of carbohydrates in the Calvin cycle. So the Calvin cycle uses the products of the light-dependent reactions, ATP, NADPH, and carbon dioxide, which is brought in, it's not a product, and produces carbohydrates. And so there you go, in that blue box there, we see the ATP coming in, we see the NADPH, again, those are coming in from the light-dependent reactions. There's our CO2 coming in from the atmosphere, which I'm going to allude to here in another bullet, and down there at the bottom, there's our sugars. And so what's the ultimate goal of the Calvin cycle reaction? I kind of already gave it away. Make organic products that plants need using the products from the light reactions of photosynthesis. So down there at that bottom of that blue arrow, there's your sugars, because plants need those organic products as well. And so where do plants and other organisms mainly get their carbon dioxide from? I already gave it away. I answered it in the first bullet. They get it from their carbon dioxide from the environment. And so again, looking at this picture, as long as you know the products and reactants, what's going in, what's going out, will be very helpful. So let's do some practice. So lesson, skill, focus, argumentation. So we're going to support a claim with evidence. Again, evidence is key. We're going to support that from biological principles, concepts, processes, and or data. So here is a multiple choice question, argumentation. A researcher claims that a certain herbicide suppresses plant growth by inhibiting chloroplast function. To test the claim, we're testing something, the researcher treats isolated chloroplasts with increasing concentrations of the herbicide. The data from the experiment are presented in Table 1. I'm not going to go over Table 1 with you. You can read that. So here's the question. Which of the following statements best describes, and again, describes the results in Table 1? You can go through and read options A, B, C, and D, pause the video, and then when we come back, we're going to see how you did. And we're back. I'm a little too excited, but we're back. And let's see, how many had A? I'm in my classroom again by myself. A's, I'll pretend my students are here, okay. B's, okay, maybe, all right. C, couple of you. D, correct answer is B. ATP synthase activity depends on a proton gradient across the thylakoid membrane. Gold star, pat on the back, you got it right. Maybe if you struggle a little bit, you know, rewind, back up this video a little bit. But again, ultimately, it's about the ATP synthase. So what should we take away? The key takeaways, and again, there was a lot of information in this video, is that chlorophyll captures energy from sunlight and excites those electrons. It's all about the electrons. As the electrons pass through that ETC or electron transport chain, protons are actively transported across a membrane. They establish a gradient, and then those protons will diffuse through ATP synthase, that enzyme powering ATP synthesis, producing more ATP. The ATP, NADPH, and CO2 are used during the Calvin cycle to produce carbohydrates through a series of reactions. So as I mentioned before, the products now become the reactants. So again, I want to thank you for your time. I want to again thank all you AP biology teachers out there working so hard to get your kids ready. And again, this is Chris Monsoor. I'm signing off from room 102 in Tiffin Columbian High School. Have a great day, and I will see you in a future video. And I'm back. Good morning, good afternoon, good evening, AP Biology, and welcome to AP Biology Daily. This is topic 3.5, cellular respiration. And this is daily video 1. My name is Chris Monsoor, and I'm coming to you from room 102 at Tiffin Columbian High School in beautiful downtown Tiffin, Ohio. In today's daily video 1, this lesson will describe the process that allows organisms to use energy stored in biological molecules. So now we're going to focus on us. It's all about us now, how we use energy. And the skill that we're going to focus on is constructing a graph, plot, or a chart, which is very important. And so what will we learn? Going to learn a lot of good stuff. But first, what processes do all forms of life use to produce ATP? How do eukaryotes coordinate cellular respiration? And how is an electrochemical proton gradient formed during cell respiration? Again, might be a good little pre-assessment to pause the video and try to jot down some answers. So fermentation and cell respiration are processes that allow organisms to use energy stored in biological molecules. You may have heard of these terms before. And so cellular respiration and fermentation are characteristics of all life forms. All living things have to use energy in one form or another. Cellular respiration and fermentation release chemical energy from organic molecules like glucose. And as you see in my image there, we have glucose as the starting point. And so oxygen, though, is not used during the process of fermentation, but is used during the process of cellular respiration. So if you look on my image there, you can see where I put a red box around oxygen is not present. And then oxygen is present. So it's kind of like you get to a fork in the road. Oxygen not present, oxygen present, which way do we go? And the one key thing here is that fermentation and anaerobic respiration are not the same process. Fermentation, the products are lactic acid or ethanol, where in anaerobic respiration, we still have pyruvate. So that's just kind of a key point I wanted to point out there. So again, what I tell my students is draw the picture. So maybe you may want to pause the video here, draw this picture, maybe add notes to it as we go. Again, use these videos as you see fit. So let's talk about cell respiration a little bit. Cell respiration, eukaryotes, we're eukaryotes, right? So we're doing cell respiration now. It involves a series of coordinated enzyme catalyzed reactions that capture energy from biological molecules. There's a lot of words in that sentence. Maybe if there's some terms you're not sure of, pause the video and define them. So I have a picture here of cell respiration, a quick overview. Again, maybe stop and draw, may be helpful. So cellular respiration involves the release of chemical energy through the breakdown of glucose and creates an energy storing molecule called ATP. Again, ATP is ultimately what we want to produce. And so what cell respiration does, it takes this chemical energy through the breakdown of glucose to create more ATP. So ATP is used by all cells to do biological work. So biological work to transport materials, for movement, for reproduction, whatever it may be, ATP is used by cells to do work. And cell respiration involves multiple metabolic pathways. We're back to those pathways again. Pathways are important, right? They're sequential so that you can release small amounts of energy each time. And so our first one is glycolysis, which occurs in the cytoplasm. So you can see where that arrow just popped in. That's glycolysis. All of these processes start with glycolysis. Glycolysis is a conserved process. All living things do glycolysis. Pyruvate oxidation. All right, pyruvate, right? Which is one of the products of a reaction in oxidation. You're losing an electron. That occurs in the mitochondria. So you can kind of see where I put that red bracket in. And then there's the arrow pointing to that process. The Krebs cycle, also known as the citric acid cycle, occurs in the mitochondria. And so there's your Krebs cycle, also known as the citric acid cycle. And then finally, we have the electron transport chain, which occurs in the mitochondria. So again, a lot of steps here, a lot of small pathways. I would stop, draw the picture, put glycolysis, pyruvate oxidation, Krebs cycle, electron transport. And so let's talk about electron transport first. So electron transport chain transfers energy from electrons in a series of coupled reactions. And so essentially what the electron transport chain does, the reactions occur in membranes of chloroplast and mitochondria and in the cell membranes of prokaryotes. And so there again, in that blue box, I'm showing you electron transport chain. So an electron transport chain facilitates, again, facilitate means to help, a series of coupled reactions used during cell respiration. Again, if you're not sure what coupled means, maybe pause the video, maybe ask a friend, put a text in the Zoom chat. And so the electron transport chains allows for more controlled and efficient transfer of energy. When I talked about those biological pathways before, you want these small series of steps. It's more efficient. And then the electron transport chain uses electron energy to establish, again, an electrochemical proton gradient across a membrane. Lots of words. Again, don't get overwhelmed. Pause the video, draw the picture, go through and figure out ways to define these. But the point of the electron transport chain is to create this electrochemical proton gradient. And there it is. There's that creating that gradient as those protons are moving across. And so what will happen is those electrons are delivered by electron carriers called NADH and FADH2 to electron transport chain. So again, lots of information here. Break it down to smaller steps. But in that green box down there, you can see where these carrier molecules come in. So again, pause and draw, you know, kind of label these pictures because there's a lot going on. And then finally in this step, ATP synthase, again, that enzyme, we know it's an enzyme because it ends in ACE, uses the electrochemical proton gradient to synthesize ATP. So ultimately, we're making some ATP. So again, a lot going on in this picture. Pause, draw it, do your own colors, label it. But again, I think we learn really well from drawing out these processes. So let's practice a little bit. There was a lot there. This is going to, so in this lesson skill, it's representing and describing data. We're going to construct a graph, plot, or chart. And often you have to be able to interpret graphs and charts, or often you have to draw them. So here is our data. We're going to representing and describing data. Yeast cells are placed in an apparatus with solution of sugar, which is a major nutrient for yeast metabolism. The apparatus detects bubbles of gas released by yeast cells. The rate of respiration varies with the surrounding temperature as indicated by the data below. So you have temperature, you have number of bubbles of gas. I'm not going to go through and read those for you. Pause the video. Graph the results represented in this data table. So this is where I would pause, do your graph, because in the next slide, I'm going to give you the answer. So go ahead, pause the video, draw out your graph. You know, if you're doing this in class, hold them up, show your friends. Maybe you need to put something on the refrigerator for mom and dad or whoever you live with. All right. So here is what a graph which would be acceptable. So again, the graph setup must contain a title, a legend, and a y-axis with bubbles of gas per minute along the bottom. X-axis temperature in degrees Celsius, correct measurement units and scaling for that axis. The data plotted, correctly plotted points in proper orientation. Points may or may not be connected with a line. Again, there's multiple ways where this graph can be represented, but it has to have those basic must contains. So again, pause the video, look at your graph, compare it to this graph, see what you did, maybe what you missed. This is a good time for discussion. So what should we take away? Key takeaways: all living things use fermentation and cellular respiration to produce ATP. All living things need energy, right? And so all living things have to access energy in a way that it is sequential and that it is not being wasted. Eukaryotes, like yourself, coordinate cell respiration in 3 metabolic stages. And again, you can go back maybe a minute or two in the video. Glycolysis, which all living organisms do. Pyruvate oxidation, again, oxidation, losing electron, and Krebs cycle, which kind of go together, and oxidative phosphorylation. And then finally, NADH and FADH2 deliver electrons to electron transport chain, which uses the electron energy to create an electrochemical gradient of protons. A lot of terminology. You may feel overwhelmed. You'll be all right. It's all right. So pause the video, go through and maybe jot down the terms you're not sure of. Again, use that Zoom chat to talk to each other, reach out to somebody, but these are your key takeaways. Go back to your pictures, all right? You got this. I believe in you. So again, I want to thank you, all you AP Biology teachers. I know you're working hard. All you AP Biology students are working hard. And again, this is Mr. Mansour. I'm signing off from room 102 in Tiffin Columbian High School in beautiful downtown Tiffin, Ohio. And this was your AP Biology Daily. And I'm back. Good morning, good afternoon, good evening, AP Biology, and welcome to topic 3.5, cellular respiration. My name is Chris Mansour, and I'm coming to you from room 102 in Tiffin Columbian High School in beautiful downtown Tiffin, Ohio. In today's daily video 2, this lesson will describe the processes that allow organisms to use energy stored in biological molecules. So again, how are you using energy from the materials that you eat? So what will we learn? Always a good question. So what will we learn? There's a lot going on in today's video. What is the function of the electron transport chain? Great question. What is the role of NADH and FADH2? There's our carrier molecules, which we've discussed previously. How does the electrochemical proton gradient power ATP synthesis? What is oxidative phosphorylation? And what is the importance of decoupling during respiration? And so let's start with the electron transport chain. And again, the electron transport chain reactions occur in chloroplasts, mitochondria, and the plasma membrane of some cells. And so the highly complex organization of living cells and living system relies on a constant input of energy. We constantly have to have energy coming in. And so what the electron transport chain does, those reactions are a conserved process, which means that all living things have some form of an electron transport chain. And so in eukaryotic cells, electron transport chains are located in the inner mitochondrial membrane and the internal membrane of the chloroplast. And so again, you have to have this membrane present to create a gradient. And so there again, in the box that just appeared is isolating that electron transport chain. And so again, there's the chain, there's your membrane. And so in prokaryotic cells, electron transport chains are located in the plasma membrane. So again, this is one of those conserved processes. So talking about the electron transport chain, let's talk about what it does. The electron transport chain transfers energy from electrons. And so membrane proteins make up the electron transport chain. And those are those things that are in blue that I just put the box around. And so electron transport chain proteins facilitate or help a series of coupled reactions, which we've talked about, using the energy from the electrons. And so what happens is these high energy electrons are donated by our electron carriers, our friends NADH and FADH2, which I will isolate here in a second. And what happens is the active transport of protons or those hydrogen ions across during electron transport chain reactions, again, trying to create that gradient. And so active transport of protons establishes that electrochemical gradient across the membrane. And so again, a lot going on here. Before I go to that last bullet, maybe draw the picture, maybe write some notes down. But the main point here is that the electrochemical gradient, again, a gradient, you have a different concentration, are maintained as a result of biological membrane impermeability to charged molecules and ions. There's a lot going on here. So if you just pause the video, the whole point is that we're trying to see that the electron transport chain transfers those electrons, and as it transfers those electrons, it allows for the formation of that gradient. A lot there. So the flow of protons, our next topic, by chemiosmosis through ATP synthase drives ATP synthesis. Again, maybe some new terms, jot down, okay, what's chemiosmosis? Put it in the Zoom chat. Okay, you should remember what ATP synthase is. I know it's an enzyme. And so the process of making ATP using the stored energy of the proton gradient is referred to oxidative phosphorylation. So oxidative, right, dealing with electrons, and phosphorylation is with your phosphates. And so NADH and FADH2, those carrier molecules, right, they lose high energy electrons to the electron transport chain, and that's oxidation. LEO, lose electrons, oxidize. And so ATP synthase, that enzyme, adds an inorganic phosphate to ADP, resulting in an ATP molecule. ADP diphosphate, resulting in ATP triphosphate. That's your phosphorylation. So you have the oxidation part with losing the electrons, and you have the addition of an inorganic phosphate. There's your phosphorylation. Again, pause the video, jot down the terms you're not sure of, and ask your teacher, or again, draw the picture and draw it out. So what happens is, in those protons moving along the gradient through diffusion, through ATP synthase, powers ATP synthase. And so we have oxidative phosphorylation, and now as those protons move along that gradient, they're going to diffuse through ATP synthase. They're all the way on the end, and as they diffuse through that synthase, they're going to allow for the production of ATP synthesis. And so there's that arrow showing you which way those protons are going to move. And so kapow, we got ATP. Maybe not quite that dramatic, but you get the idea. So let's move on a little bit. And so in cell respiration, decoupling, right? So we have a coupled reaction. Now we're going to separate the reactions. So the decoupling oxidative phosphorylation from the electron transport chain generates heat. And so energy is stored in proton gradients. And so that's where the energy is stored. In decoupling oxidative phosphorylation, a lot of terms here, again, pause the video. Decoupling or separating oxidative phosphorylation from the electron transport chain refers to the proton gradient not being used by ATP synthase to produce ATP. A lot going on there, all right? So we're not being used by ATP synthase. So we're decoupling or taking the process apart. And so when decoupling occurs, the energy stored in the gradient is released as heat. And that heat from decoupling, right, separating the processes, can be used by endotherms like us, right? Endothermic organisms to regulate body temperature. Again, a lot of terms going on there. Pause the video, right? Go back to the previous slide, draw out the picture. But the key point there is that when we decouple those processes, when we decouple oxidative phosphorylation, we release some heat that can be used to regulate body temperature. So what should we take away? Key takeaways, there's a lot here. So one, the electron transport chain allows for efficient transfer of energy from electrons to proton gradient. So again, that efficient transfer. Our carrier molecules, NADH and FADH2, donate these electrons to electron transport chain. And then by donating these electrons, they help the electrochemical gradient provide stored energy that is transformed by chemiosmosis used by ATP synthase to synthesize ATP. A lot going on there. Chemiosmosis, those protons are moving through, and as they move through, ATP will be formed. Oxidative phosphorylation is a pathway utilizing electron transport chains, chemiosmosis, ATP synthase to make ATP. I would go back a couple slides, draw that picture, and just label it with all these terms and try to explain it to yourself or explain it to somebody else. And finally, when decoupling occurs during cell respiration, heat is released that can be used for thermoregulation, endotherms like us. I know there's a lot of information in here. Pause the video, take your key takeaways, draw a picture, right, annotate it. But again, by practicing with this terminology, it'll hopefully stick. So again, I want to thank you. Again, thanks to all the AP Biology teachers, AP Biology students. Thank you for sharing your time with me. This is Chris Mansour signing off from room 102 at Tiffin Columbian High School in downtown Tiffin, Ohio. And have a great day, and I'll see you in a future video. Good morning, good afternoon, good evening, AP Biology. My name is Chris Mansour, and I'm coming to you from room 102 in Tiffin Columbian High School, and this is AP Biology Daily, topic 3.5, cell respiration. In today's video, daily video 3, this lesson will describe the process that allows organisms like you to use energy stored in biological molecules. And so what will we learn? Always a great question. So what will we learn? What is the purpose of glycolysis? What is the fate of pyruvate? What are the products of the Krebs cycle / citric acid cycle? What happens to the electrons extracted in glycolysis and Krebs cycle, or what we also know as the citric acid cycle? How does fermentation differ from aerobic cell respiration? And finally, how is energy released from ATP? Again, I would pause the video, jot down some answers, try to see, do a little pre-assessment, see how you do. So let's talk about glycolysis first. So glycolysis is a biochemical pathway that releases energy stored in glucose. So I have an image here. You may want to draw a picture of it and maybe annotate it as we go. So glycolysis results in the production of pyruvate, NADH, and ATP. Again, NADH being a carrier molecule, ATP, right, is going to be used for energy, and then we have our pyruvate. And so there's our pyruvate there in the middle in that red box. As you can see, glucose to glycolysis to pyruvate. So let's talk about pyruvate a little bit more. So pyruvate is transported from the cytosol to the mitochondrion. And so pyruvate is actively transported, all right? So active transport going against the gradient through the mitochondrial membranes into the matrix. There's a lot of M's in that sentence. And so then the pyruvate is oxidized, right? We talk about oxidation, losing electrons, and the product of that pyruvate oxidation then can enter the Krebs cycle. Again, I would draw this picture out, annotate it. So around that black box there, we're showing pyruvate, right? We're showing it's being oxidized, and then we have its new product. So let's talk about the Krebs cycle now, or what's sometimes called the citric acid cycle. In the Krebs cycle, carbon dioxide is released from organic intermediates. And so what the Krebs cycle is, it's a pathway that involves many key reactions. And so the first being carbon dioxide is released from intermediate reactions. And so you can see right there, there is that CO2 being released. Also, high energy electrons are transferred to NADH and FADH2. Those are your electron carriers. And so there's the NADH there, and there's your FADH2 there. And then also ADP is phosphorylated, forming ATP. And so again, we have CO2 being released, you have high energy electrons being transferred to NADH and FADH2, and you have ADP phosphorylated, forming ATP. So let's talk about those electrons. So the electrons that were extracted in glycolysis in the Krebs cycle are going to be transferred to the electron transport chain. And so the NADH created in glycolysis and the NADH and FADH2, again, trying to keep those straight, creating the Krebs cycle, donate electrons to the electron transport chain. So there you are. There's your carrier molecules. There's the NADH coming in. There's your FADH2. Those electrons then are transferred between membranes, proteins, and electron transport chain, which we've talked about. There's that blue box highlighting the electron transport chain. And then the electron transport chain is going to establish an electrochemical gradient of protons, hydrogen ions, across the inner mitochondrial membrane. Again, we have to create that gradient. And so there are H pluses moving across, and they're going to create that gradient. And so let's talk about fermentation a little bit now. So fermentation allows glycolysis to proceed in the absence of oxygen. And so what will happen there is that the products of fermentation, like I said, are in a previous video, are different than the products of anaerobic respiration, because the products of fermentation are ethanol and lactic acid. Those are the byproducts. And so there they are, as I highlighted there in the red box. So lactic acid or ethanol. And so let's also talk about this conversion of ATP to ADP. So the conversion of ATP to ADP releases energy. So in that picture, you can see where that ATP is giving off that phosphate. So energy is released when the chemical bonds are broken. And so when that bond is broken between the second and third phosphate, that's when you release energy. And that energy released from that ATP hydrolysis can be used to power many metabolic processes such as movement, transport, reproduction, whatever your body needs for energy, it's going to be able to acquire it from that ATP hydrolysis. And so there I have isolated that process. You've got your ATP with the 3 phosphates, you release a phosphate, energy is released for use by the cell. And so what should we take away? Great question. A lot of key takeaways here. Again, I would pause the video and maybe see how your initial questions match up with the key takeaways. Glycolysis releases energy and glucose to form ATP and NADPH. So again, glycolysis is a process that all living things do. That pyruvate is transported to the mitochondria where it's oxidized, and again, oxidized, losing some electrons. During the Krebs cycle or the citric acid cycle, carbon dioxide is released. So that's when we're actually, when CO2 is released, is during the Krebs cycle. ATP is synthesized, and NADH and FADH2 are produced. A lot of things going on during that Krebs cycle, a lot of products. The electrons extracted in glycolysis in the Krebs cycle, right, or the citric acid cycle, they're transferred by NADH and FADH2 to the electron transport chain. So again, go back, maybe draw the picture and annotate it. Fermentation, again, allows glycolysis to occur without oxygen, and it produces ethanol or lactic acid. And again, if you're kind of curious about what ethanol or lactic acid are, again, pause the video, maybe put a question in the Zoom chat, or maybe just Google them. And then finally, energy is released when that third phosphate is broken off, resulting in ADP, and you release some energy. So again, I want to thank you again for your time. Thank you to your AP Biology teachers. I know you're all working very hard. AP Biology students, you're also working very hard watching these videos. Hopefully the information presented will be helpful to you. And again, my name is Chris Mansour. I'm signing off from room 102 in Tiffin Columbian High School in Tiffin, Ohio. And have a great day, and I'll see you in a future video.